All right, we've already talked about the cell cycle, the process of mitosis. Now we're going to get into meiosis and sexual reproduction. In this chapter, we're going to cover an overview of meiosis, genetic variation, the phases of meiosis, and then how meiosis compares to mitosis, finally the cycle of life, and how changes in chromosome number and structure can ultimately affect the organism. But we're going to start off by just doing a very general overview of meiosis and mitosis. Meiosis is, like mitosis, is a cellular process of reproduction and division. However, unlike mitosis, which is primarily used for growth and tissue replacement, meiosis is going to be used only for sexual reproduction. This is the process whereby an organism can scramble their DNA and reproduce offspring that is going to be genetically different. In mitosis, the resulting cells are going to be called daughter cells. In meiosis, the resulting cells are called gametes. These are also called sex cells, and specifically they're going to be egg and sperm cells in animals. In mitosis, the daughter cells are going to be genetically identical to the parent, since what we want to do is grow and replace. So like if I scraped my knee and I needed to build more skin cells, I would want those skin cells to be exactly identical to the skin cells that were previously there. Do not want genetic variation grown out of my knee. Not ideal. So in the case of mitosis, daughter cells are going to be genetically identical to the parent cells they come from. In gametes, however, gametes are going to be genetically different from the parent cells. Like mitosis, the original parent cells are diploid. So in mitosis, we start off with diploid parent cells and we end up with diploid daughter cells. Now would be a great time for you to review what does diploid mean. However, unlike mitosis, where the daughter cells each receive a genetic copy of the parent DNA, in meiosis, each gamete is only going to receive half the parent's DNA, making them haploid. So think about it. What benefit of each gamete only receiving half of the parent's DNA? Why would we want that? Why do we do that? Why do we produce, why do diploid parent cells produce haploid gametes in meiosis? So like we said, meiosis is going to produce a haploid number, is going to produce haploid gametes. Means that they only have one of each set of chromosomes. If there were no reduction in chromosomes in meiosis, the number of chromosomes would double in each generation. In other words, if I passed 46 chromosomes on to my offspring, and my husband passed 46 chromosomes off to our offspring, we would have offspring with 92 chromosomes. Their offspring would have offspring with 184 chromosomes. The genetic material would double every single generation, which would be just pure insanity. So instead what we do is when we are producing sex cells, when we're producing gametes, we're going to have the number of chromosomes, so that way the offspring can receive one set of chromosomes from each parent. So this is beneficial because, as I said, there's going to be no reduction of chromosomes in meiosis. If there was no reduction of chromosomes in meiosis, the number of chromosomes in the organism would double with every generation. So to produce the next generation, the haploid gametes have to fuse in fertilization to form a diploid in order to form a diploid organism. At the point where those two gametes fuse, egg and sperm meet, we're going to form what is known as a zygote. So this is post-fertilization. When the cell becomes diploid, it is known as a zygote. That zygote is officially diploid, has a completed set of chromosomes, and is going to be our next generation. So another thing that we would like to point out, or as we mentioned previously, chromosomes are going to occur in pairs. So for example, in humans, we have 23 different types of chromosomes. But as you can see here, chromosomes tend to occur in pairs. So we have 46 different chromosomes in our body. We have 23 different types, but we have 46 total because we have 2 of every type of chromosome. So just like twins in humans, these pairs of chromosomes tend to look alike. They tend to be the same length. They tend to have the same positioning. The centromeres are going to be located in the same relative position. When they're stained, they're going to show the same banding patterns. And the reason for this is that these homologous chromosomes, or that's what we call it. So just like a twin, we call these homologous chromosomes, meaning that they code for the same type of genetic information. Homologous chromosomes code for the same genes. So for example, we have 2 sex chromosomes. Down here in the corner, we have 2 sex chromosomes. One sex chromosome comes from your mom, one sex chromosome comes from your dad. If you're a female, you got an X chromosome from each parent. If you're a male, you received an X chromosome from your mom and a Y chromosome from your dad. Now, both of these chromosomes code for the sex of an individual, but they are 2 different chromosomes. For another example, chromosome number 1 over here. Maybe at this banding pattern right here, we're coding for height. So it could be that you get a really tall gene from your mom and a really short gene from your dad. Both of these genes, they code for height, but they may be different chromosome, or they may be different genetic information. Oh, yep, there you go. They're going to code for similar genes. So in this case, both of these genes are going to code for height. Or over here, we might have genetic information for hair color. So you might have brown hair from your mom and blonde hair from your dad. Both of these code for hair color. They're going to be found in the relatively same location, but it might be slightly different genetic information. So here's just another example or another diagram showing homologous pairs and what those homologous pairs are going to look like during the process of meiosis. So while homologous chromosomes code for the same trait, they may have differing genetic instructions for that trait. Like I said, for example, this chromosome may be responsible for the length of your finger. Your paternal chromosomes may code for long fingers, while maternal chromosomes code for short fingers. This variation of gene types is called an allele. And I know I don't have that term up here, but we're going to spend a lot of time looking at alleles later in this chapter and throughout our next chapter. So here's just some of the words, so that way you've got some notes that you can put down to accompany those previous slides. Homologous chromosomes. Many genes exist in several variant forms in a large population. The variants that exist for a gene are called alleles. In other words, if we are coding for if we're coding for eye color, brown eyes might be an allele, blue eyes might be an allele, green eyes might be an allele. Those are different kinds of information for the same type of information, or different variations for the same type of information. We're all coding for eye color, but we just may have different variations for it. Those would be the alleles. Now, an individual may have identical alleles for a specific gene on both homologs. In other words, they're homozygous for the trait. For example, you might receive a trait for long fingers from your mom and long fingers from your dad. In that case, you would be homozygous for the trait of finger length. You may also be heterozygous, meaning that the maternal allele differs from the corresponding paternal allele. You may have long fingers from your mom and short fingers from your dad. That would mean that you are heterozygous for the trait. You have 2 different alleles for that particular trait. Oh, there you go. For example, the gene coding for short fingers on one homolog and a gene coding for long fingers at the same location on the other homolog. So here's just sort of an overview of meiosis. Meiosis is going to be, or is often referred to as reduction division, meaning that we're starting off as a diploid cell and we're going to end up as a haploid cell. In order to do this, we're going to need to undergo 2 different nuclear divisions, and we're ultimately going to be producing 4 haploid daughter cells. 2 nuclear divisions occur in 2 stages, so we're going to divide the process of meiosis into 2 categories, meiosis 1 and meiosis 2. In meiosis 1, we're going to undergo DNA replication. At this point, the very first thing that the cell has to do prior to undergoing its first nuclear division is replicate our genetic material. Just like in mitosis, the parent chromosomes are going to replicate to form sister chromatids attached together at the centromere. However, this time, instead of the sister chromatids lining up with their centromere down the middle of the cell, the sister chromatids are going to find their homologous sister chromatids and they're going to start to partner up. This process is what we call a synapse. So if we're talking about your chromosome that codes for finger length, your maternal chromosomes that code for short fingers will find and stand next to, or synapse, with your chromosomes from your dad that code for long fingers. So our homologous chromosomes are going to find their buddy and pair up. And basically they're all going to pair up in their homologous pairs and migrate as a couple to the center of the cell. Once we get to the center of the cell, the members of each pair are going to separate. This separation means that only one of the duplicated chromosomes from each homologous pair makes it into the daughter cells. Why would we want this? What benefit would this be? Why do we go through all the trouble to line the chromosomes up in pairs if we're just going to be separated again in a moment? Well, the reason that we would do this is because we want to reduce the number of chromosomes. When we started off here, prior to DNA replication, we had 4 chromosomes. Now, after we've undergone our original or our meiosis 1, we now only have 2 chromosomes in each cell. Now, we have duplicates, we have sisters that we're still going to have to be divided, but we only have 1 of each type of chromosome. So if the short chromosomes code for finger length and the tall chromosomes code for hair color, now each offspring has 1 chromosome for finger length and 1 chromosome for hair color. Once completed, once we have finished, we still only have 2 cells, and what we're aiming for is 4 cells that only have 1 copy of each chromosome. So at this point, we move into our second nuclear division, and this process follows very closely to that of mitotic division. The sister chromatids are going to line up along the metaphyseal plate, and they're going to be separated by the mitotic spindle fibers. We're going to go through all of the same stages of prophase, metaphase, anaphase, and telophase. The sister chromatids are going to separate, and we're going to ultimately end up with 4 haploid cells. Now, quick question for you. Do any one of these cells, 1, 2, 3, 4, any one of these cells match the original parent cell? No, they don't. They don't. The original parent cell was diploid. The original parent cell had 4 chromosomes, 2 coding for finger length, 2 coding for hair color, and each one of these chromosomes each one of these daughter cells only has 1 of each type. So their genetic variation is going to be a little bit different. They only have half the original genetic material. So this is where genetic variation is going to really come into play, and we're going to talk through it. There are a lot of different types of genetic variation that can happen throughout this process of meiosis.